In this presentation, a novel immunotherapeutic nanoparticle system developed in our lab for the treatment of cancer will be discussed. Before beginning, our team would like to acknowledge the Northern Cancer Foundation for funding this research. Cancer immunotherapy is an alternative strategy for cancer treatment which focuses on modulating the immune system to fight cancer. This opposes traditional treatments that directly target the cancer cells, including surgery, radiotherapy, and chemotherapy. Our system was developed based on the capacity of cytotoxic T lymphocytes, or CTL, to kill and generate long-lasting memory against cancer cells. In brief, the immune system can recognize tumor cells because they release tumor-associated antigens, or TAA, which are recognized by antigen-presenting cells, or APC, including dendritic cells, or DC. DC present the antigens to CTL, which in turn recognize and kill the cancer cells, a process heavily mediated by the cytokine interferon gamma, or IFN gamma. However, tumors have developed ways to inhibit immune cell killing via an immunosuppressive tumor microenvironment, or TME, which is in part composed of tumor-associated macrophages. In order to kill cancer cells via the immune system, we need to modulate the TME, which can partially be achieved by its reprogramming to a pro-inflammatory M1-like phenotype. In order to achieve those goals, our lab has developed Ara-C, a cationic glycoliposome containing muramyl dipeptide, termed DS, and low doses of the chemotherapeutic cytarabine, or Ara-C. The liposome was designed to improve IFN gamma-positive CTL responses and APC targeting, while MDP is added as an immunomodulator that can activate APC. Ara-C was also designed to be used in combination with TAA in order to improve tumor recognition and CTL targeting. Taken together, Ara-C was designed to stimulate the immune system, revert immunosuppression, and improve long-term memory of CTL against cancer cells. In this presentation, I will further discuss preliminary data showing that Ara-C offers interesting anti-cancer immune properties and warrants further studies. To give some context behind incorporating Ara-C and DS, the system was first investigated during a collaboration study with the lab of Dr. William Stanford at OHRI in Ottawa. The goal was to improve the stability of Ara-C, which greatly limits its clinical use as a chemotherapeutic. During this study, we were able to show Ara-C stability is greatly improved upwards of 48 hours when entrapped in the glycoliposome, with approximately 90% drug entrapment. Subsequently, during a screening to identify chemotherapeutic drugs with immune properties, Ara-C alongside a series of chemotherapeutics were entrapped at low, non-toxic doses in DS and incubated with peripheral blood mononuclear cells, or PBMC, which are composed of various immune cells isolated from the blood. Surprisingly, Ara-C greatly improved the activity of DS, with Ara-C producing the highest IFN gamma. This led to the investigation of the Ara-C system as an anti-cancer immunotherapeutic system. Prior to further investigations of Ara-C, we first confirmed that the system was safe. The system did not induce hemolysis of red blood cells and was not cytotoxic to PBMC. The system was also shown to be non-toxic in mice, with weights being maintained in Ara-C vaccinated mice, without any fever being induced 24 hours post-injection. We then analyzed the cytokine profile of Ara-C using PBMC isolated from healthy volunteers after 6 days of ex vivo incubation. Although variability exists between donors, Ara-C consistently and significantly increased the production of IFN gamma, while Ara-C or DS alone did not, as compared to the non-treated control. Importantly, while DS increased the unwanted immunosuppressive IL-10, Ara-C did not. It is also important to note that although the pro-inflammatory cytokines IL-6, TNF-alpha, and IL-12p40 are not higher in the Ara-C treatment at 6 days, those cytokines are increased above baseline at earlier time points, indicating a potential shift to the adaptive immunity after 6 days. Preliminary data also suggests that the response observed for healthy individuals is similar in colorectal cancer patients pre-chemo. However, this response is almost entirely lost in cancer patients after repeated exposure to high-dose chemo. This is not surprising as high-dose chemo is associated with immune suppression, suggesting that such treatment is highly toxic and may prevent proper immune functions necessary for long-term clearance of cancer cells and prevention of cancer recurrence. The cytokine profile observed in PBMC led to the exploration of the role of Ara-C in activating CTL in the presence of antigens. In order to investigate this, we used a Ceph peptide pool derived from common viruses. This pool allows the rapid testing of our system using healthy donors, as most people are expected to respond to the viruses found in the pool due to previous viral exposure. This experiment served to prove that Ara-C could induce an improved memory response in the presence of TAA, which are recognized in a similar way as Ceph in the case of cancer patients. Here, we were able to show that in 3 different donors, Ara-C improved the alpha-gamma positive CTL response to the peptide antigens. Furthermore, the system decreased the variability of responses between the donors as compared to Ceph-stimulated cells without Ara-C. This data supports that TA could be used in combination with Ara-C to help direct the response against cancer cells, thereby inducing long-term memory. Thus far, we have shown that Ara-C displays promising immunostimulatory activity. However, immunotherapeutics have been known to fail clinically due to the immunosuppressive TME preventing CTL-mediated killing. As a preliminary assessment of macrophage immunomodulation, we differentiated primary human monocytes to anti-inflammatory M2-like macrophages in vitro with MCSF. 5 days post-differentiation, cells were left untreated, treated with MCSF, or treated with Ara-C. Surface marker CD163 associated with an M2 phenotype and CD80 associated with an M1 phenotype were assessed by flow cytometry. Ara-C was shown to increase CD80 and decrease CD163, indicating a potential for reprogramming of macrophages. Finally, early mechanistic studies were conducted to elucidate the mechanism of Ara-C. We first investigated the activation of TLR4, a common target of immune adjuvants associated with the induction of pro-inflammatory cytokines. Using the TLR4 inhibitor CLI-095, we showed that Ara-C activity in DC is not mediated through TLR4. The TLR4 agonist glucopyranonyl lipid A, GLA, was used as a control to show that CLI-095 inhibits TLR4. This data further demonstrated that Ara-C is immunostimulatory and increased IL-12, IL-6, and TNF-alpha in DC at an early 24-hour time point. Upon discovering that Ara-C is not linked with TLR4, we analyzed activation of the STING, TPK1, IRF3 pathway by analyzing phosphor-IRF3 by Western blot analysis of THP1s, a human monocyte cell line. STING, or the stimulator of interferon genes, is an interesting target in cancer immunotherapy, as it can enhance immune responses through the induction of type 1 interferons while inducing innate and adaptive immune responses through the maturation of DC and the induction of CD4 T cells and CTL responses. We were able to show that although weak, Ara-C activated IRF3 and DS maintained the activity of the STING agonist 2,3-CJMP even when using a 10-time lower dose as compared to the agonist alone. These data suggest that Ara-C can activate the STING pathway, and DS can synergize with STING agonists. To conclude, we have developed a multi-targeted system with potential to be used as a cancer immunotherapeutic. Our system has already shown to stabilize Ara-C and may reprogram cells in the TME, as evidenced by an increase in CD80 and a decrease in CD163 in macrophages. The system also targets healthy cells by inducing an anti-tumor immune response with an initial increase in IL-12, IL-6, and TNF-alpha, followed by the production of alpha-gamma, without production of the immunosuppressive IL-10. This could be linked to the activation of STING, but not TLR4. Finally, CTL responses were improved in the presence of target antigens. Although promising, the Ara-C must now be further investigated, especially within the scope of suppressing the TME and increasing recognition of tumor cells by CTL. In order to do so, further investigations will test the efficacy of the system in vivo using cancer mouse models. Furthermore, the combination of Ara-C with different cancer treatments that also display potential as immune modulators, such as low-dose radiation therapy, will be explored.